So last time we learned about principles of Mendelian inheritance, and these serve as a baseline default for how we can expect traits to be inherited. So if you take a trait and you do a certain cross, you look at progeny, you should be able to predict how many of those progeny should have the trait, for example. However, in biology, there are exceptions to every rule. So in the next couple lectures, for this lecture and the next lecture, we're going to talk about why different traits might not follow Mendelian rules. And one common cause of non-Mendelian inheritance is sex linkage. And what this means is that genes are located on the sex chromosomes, on the X or the Y chromosome. In order to talk about sex linkage, though, we first need to cover sex chromosomes, what they are, and sex determination, what makes males male and females female. So let's just take a moment to define what are males and females. So among sexually reproducing organisms, life cycles pretty much always include a diploid stage and a haploid stage. So for mammals like us, our haploid stage is our gametes. So for example, here we've got eggs and sperm. For other plants and animals, it might be more complicated. So we define males as the organisms that produce small, mobile gametes, and females produce relatively large, immotile gametes. And these are sperm for males and eggs for females. So as somebody who researches sperm for a living, I can tell you that there are actually many exceptions to that rule. Some sperm are quite large. So these are the 5.8 centimeter large sperm of the fruit fly Drosophila bifurca, shown by comparison next to one of the eggs. And this system is about as close as we get to what we might call isogamy, meaning equal-sized gametes. It's not quite there, but it's close. For most organisms, though, we're going to say that males produce tiny sperm and females produce large eggs. Many organisms are both male and female, make both eggs and sperm within a single individual. These are hermaphrodites, and we refer to these types of organisms as monoecious, meaning from the Greek for one house, whereas individuals who have sexes separated in organisms that have sexes separated in different individuals are what we call dioecious, meaning for two houses. So among dioecious species, there are several ways to be male or female, and these largely fall under either genetic or environmental mechanisms, and we refer to the genetic mechanisms as genic. And in 1891, Hermann Henking first discovered what he referred to as the X body in firebugs, and noticed that it didn't behave like the other chromosomes during meiosis. In 1901, Clarence McClung determined that the X body, he actually referred to it as the accessory body, was a chromosome. So Henking discovered this thing that he noticed is inherited differently, and Clarence McClung actually called it a chromosome. And he found that females had one more of these X-body or chromosomes than males do, and suggested that the X-body had something to do with sex determination. Meanwhile, in 1905, Nettie Stevens and Edmund Wilson found that in grasshoppers, females have two X's, males have one. And in other insects, they found that males also had a smaller chromosome, which they called a Y chromosome. So this is the origin of what we refer to as the X chromosome and the Y chromosome. Stevens and Wilson also showed that the X and Y chromosomes separated into different gametes during spermatogenesis. So a gamete will contain either an X or a Y, but not both. Sperm will contain either an X or a Y, but not both. But all of the eggs will contain just a single X chromosome. And then in addition to the rest of the haploid genome, of course. So when Y-bearing sperm fertilize an X-bearing egg, the resulting progeny is male, while when an X-bearing sperm fertilizes an X-bearing egg, you get a female. So the equal division of X and Y chromosomes as a result of meiosis explains the equal sex ratio. This is called chromosomal sex determination. It is a form of genic sex determination, and chromosomal sex determination occurs when the presence or absence of a sex chromosome determines gender. So in the case of many organisms, the absence of a Y will determine that that individual is a female. Sex chromosomes are essentially defined as chromosomes that determine gender, that determine sex, and they're morphologically distinct and therefore recognizable on a karyotype. It is important to note that it is not the chromosomes per se that are determining sex, it's the genes that are on those chromosomes. So chromosomal sex determination is again just another form of genic sex determination. There are some organisms that have no distinct sex chromosomes. You really can't tell their chromosomes apart, yet clearly they have different sexes. Those sexes appear to be determined genetically, but we just haven't located the exact genetic mechanisms yet. It may also be interesting for you to know that in many organisms, including fish, including a number of other vertebrates, including a number of insects, we don't very well understand exactly how sex is determined. We think it's genetic, but sometimes it could be environmental, sometimes it's a little of both. How we think of male versus female can often be a little bit blurred in nature. And so what I'm going to talk about next is just some typical examples of different mechanisms of chromosomal sex determination. So in some insects, and this includes grasshoppers and their relatives, these are called orthopterans, in cockroaches, and in this model organism, the nematode Caenorhabditis elegans. All of these animals have what's called an XX-XO system. This is also present in some mammals. And in these animals, females have 2 X's and males have 1 X. And the O here is not an actual chromosome. There's no O chromosome. It just means that males only have 1 X. So here females are what we call homogametic. They produce eggs that all have 1 X. And males are heterogametic, where they're producing gametes that have either an X or they don't have an X. So males are producing gametes that are not all the same, thus they're heterogametic, whereas females are producing gametes that are all the same in terms of the number of sex chromosomes and the type of sex chromosomes that they have. So that makes the females homogametic in this case. Many animals, including us, most mammals, some insects, have females that are XX, same as before, and again these females are homogametic. And the male has an X chromosome, but they also have a smaller chromosome, which is the Y. And the X and Y chromosomes pair during meiosis with the help of these pseudo-autosomal regions, which are in red. And these are the very small regions that are homologous between the X and the Y, because evolutionarily, the Y is originated from an ancestral X chromosome. And in some fish, some crustaceans, birds, lots of reptiles, and lots of butterflies and moths, females are what we call ZW, and males are ZZ. So in this case, the females are the heterogametic sex, and the males are homogametic. And similarly to the XY system, the W chromosome is a much smaller chromosome than the Z chromosome is, similar to how the Y chromosome is much smaller than the X. And I believe it is understood that the W has evolved from the Y. Now, it's also important to bear in mind that many of these systems of chromosomal sex determination have evolved repeatedly over the course of the history of life on Earth. And so the ZZZW system that we see in lepidopterans and butterflies and moths is not necessarily the same ZZZW system that has evolved in flatfish, which is not the same ZZZW system that evolved in birds. And so it's important to recognize that the evolution of sex chromosomes is incredibly labile and can evolve again and again and again in different groups of organisms. So this is just a practice question that if you want to pause the video and take a stab at it, go for it. Just asking, how does the heterogametic sex differ from the homogametic sex? And it is the case that the heterogametic sex is defined as the one having different sex chromosomes, so the answer is B. All right, so all of the cases that I talked about in the context of chromosomal sex determination, again, are forms of what we call gene-X sex determination. And in a more general way, gene-X sex determination is just when sex is determined by genes located somewhere, but we don't always know where they are. When they're on sex chromosomes, it's still technically gene-X sex determination, but we just, because it's somehow different, because the sex chromosomes are recognizable, we call that chromosomal sex determination to distinguish it from gene-X sex determination, which just means we don't know where the genes are. All right, so environmental sex determination is a case like that found in turtles or crocodilians, which are alligators, crocodiles, and caimans, and also a few birds, where sex is determined by the developmental temperature. So when eggs are incubated at higher temperatures, you might get more males or more females, depending on what you are. And at cooler temperatures, you might produce more females or more males. And it's not really the case that you're going to completely switch from producing all males to producing all females. It's more that the sex ratio changes at higher or lower temperatures. So what happens in at least turtles and crocodilians, they both will bury their eggs in vertical nests. And these nests are going to span the range of whatever the critical temperature is. That's sort of the set point for determining sex. And so females might be towards the bottom of the nest and males towards the top, depending, because it switches in turtles and in crocodilians, I believe. And so overall, though, within a clutch, you tend to get an equal sex ratio. Another type of environmental sex determination is not necessarily temperature dependent, but involves social environment. So one example is sequential hermaphroditism, where in some fish and invertebrates, individuals begin life as one sex, and then at some point they switch, often due to some social environmental cue. So, for example, these common slipper limpets, they'll settle on a substrate, and they'll settle in vertical stacks. And the individuals on the bottom, the first individual to settle becomes female, and then individuals who settle on top of her become male, and his sperm will fertilize her eggs, and after a time, the bottom male, once he grows bigger, will become a female and start attracting other males to settle on top of him, or now her. And so you end up with a stack of limpets where the bottom individuals are female and the topmost individuals are male. We also have clownfish or anemone fish, where the one pair, a male and a female, will occupy an anemone, and if the female gets removed, then the male becomes the female, and another male will come take his place, which puts the whole story of Finding Nemo into perspective, because really Nemo's dad should have then become his mom, and then he would have gotten a new dad. So species where individuals are female first, as in here, are I'm going to say protogynous, or protogynous, whereas individuals who are male first and then become female are called protandrous. Okay, so some individuals or some species have multiple sex determination mechanisms. Some individuals have multiple sex determination mechanisms, some species. For example, in bearded dragons, you can have both influences on both genes and temperature can influence sex development, where females are usually ZW, but at higher temperatures, ZZ individuals who would normally be male, they'll develop as females as well. And then you can get a whole bunch of general craziness where, for example, in platypus, they have 10 sex chromosomes. They have X's and Y's. Females have 10 X's and males have 5 X's and 5 Y's. And how these guys segregate during meiosis is very interesting. So you should look that up if you want to know more. Platyfish have 3 sex chromosomes, a WX and a Y, where certain combinations of these sex chromosomes can be female, whereas other combinations are going to be male. So in humans, there are a number of sex chromosome abnormalities that lead to different types of disorders, one of which is called Turner syndrome. And these are individuals that completely lack a Y chromosome. They lack a Y, and they're what we call XO. And these individuals are phenotypically female. They often have a common characteristics. They often have a number of common characteristics that include they tend to be short in stature. They have a broad chest, a low hairline, a webbed neck. They tend to be normal in intelligence, and they also tend to be sterile. There are no known cases of individuals missing both X's, and this suggests that the X chromosome is really critical for development, such that if an embryo is missing an X chromosome, it becomes inviable and doesn't make it to birth. Another chromosomal abnormality that leads to a disorder is one that causes Klinefelter syndrome. And this is what happens when you have multiple X's. So Turner syndrome lacks a Y. Klinefelter syndrome has multiple X's. And this individual has a karyotype that includes 2 X's and a Y, but you can also have more than 2 X's, and so the XXY is the most common form. These individuals are phenotypically male. They are often tall. They have small testes. They're sterile. They have slight feminization, normal intelligence, and they can be XXXY. They can have 3 X's, 4 X's. They can also be XXYY. So we can also get 3 X's and no Y's. This is called Triplo-X. And these individuals are phenotypically female. They're often tall and thin. They have normal intelligence, and they are fertile. The Triplo-X population does have an elevated incidence of intellectual disability beyond that expected in the XX population. And the severity of this intellectual disability increases with the number of X's. So the more X's that an individual has, the more likely that they are to manifest some kind of intellectual disability. Okay, so given these observations, what can we say about the role of sex chromosomes in sex determination? So first of all, we can say that the X chromosome is essential for both males and females to properly develop. If you're missing an X entirely, you don't develop at all. If you have a Y, you're male no matter how many X's you have. So for Klinefelters, you can have 4 X's and just 1 Y and still be phenotypically male. Likewise, if you have an X but no Y, you're female even if you only have 1 X. In addition, both the X and the Y are important for fertility. At least 2 X's are needed for female fertility, and you can also have too many X's. So, let's see. So in humans, the sex determining region is called SRY and is located on the Y chromosome, and it essentially directs embryonic development towards maleness. It's a transcription factor that turns on other transcription factors that start up a whole suite of gene expression that leads to the development of male characteristics. So in the absence of this sex determining region, you end up with femaleness. And so you end up, you as an embryo, as human embryos, we generally all start out with the default of being female. And if the SRY locus is present, then we start development as males. So in 1990, the SRY was found to contain a gene that's specifically called testis determining factor. So we knew that the sex determining gene region Y was really just a region. We didn't know what was in it. We knew that it was on the Y chromosome. We knew approximately where it was, but we didn't know what gene was there. And so in 1990, they found the genes called testis determining factor, and it encodes, as I said, a transcription factor that controls and upregulates other genes that promote testis development. So rarely XX individuals can be male. So how can this be? Do I tell the answer? Sort of. Okay. So here's just the Y chromosome. We've got our this is the Y chromosome. We've got a pseudo autosomal regions. SRY is located here, and sometimes I just want to see what this is. Sometimes we get individuals that are XX, but they can be male. So what can happen is that somehow you get a chromosomal translocation event, and we'll learn more about that in a couple lectures, where the sex determining region actually gets switched, gets relocated onto one of the X's often, just because a random recombination event happened, or maybe it was translocated onto a totally different chromosome. And as long as it's present in the genome somewhere, it doesn't need to be on the Y chromosome. It could be anywhere. You still become male. So as I said earlier, genes are sex-linked if they occur on a sex chromosome. So for humans, sex-linked genes are on either the X or the Y. And since there aren't too many Y-linked genes, there aren't too many genes on the Y, most of the genes that we talk about are X-linked. So females are going to have 2 copies of X-linked genes, and they can be either homozygous or heterozygous. Males will only have 1 copy of X-linked genes, and they're what we call hemizygous, because they really only have 1 X chromosome. So you can't be heterozygous or homozygous if you only have 1 chromosome, so we call them hemizygous for X-linked genes. Males and females are going to differ in their patterns of inheritance of sex-linked genes, but these genes individually each still obey Mendelian principles. And so this phenomenon was first discovered in the fruit fly Drosophila by Thomas Hunt Morgan in 1910. And he found a random white-eyed male in one of his colonies, one of his stocks of Drosophila melanogaster, and he mated him to a wild-type female, so a wild-type red-eye female. So this is the cross that he performed. And their progeny were all wild-type, and they were all red-eyed. And what this suggested was that the white-eye, the allele that causes white-eye, was recessive. In the F2 generation, however, he ended up with different proportions of phenotypes for the different sexes. So he found that females were all wild type and males were half wild type and half white eye. And Thomas Hunt Morgan proposed that the gene for white eyes is on the X chromosome, and different patterns of inheritance for males and females is very much a hallmark of sex-linked traits. So let's see. So to demonstrate that the white eye allele was on the X chromosome, he did a reciprocal cross where he was able to generate a white-eyed female, and he crossed her with a red-eyed male, with a wild-type red-eyed male, and he was able to test some hypotheses about so based on, he was able to calculate some expected proportions that he would get based on based on the location of the, based on whether or not the white eye allele was on the X chromosome. So in the F1 generation, he found what he expected to find, which was entirely red-eyed females and entirely white-eyed males, and he crossed these individuals and found equal proportions now of red-eyed females and white-eyed females and red-eyed males and white-eyed males. And if you do the, look at the, calculate the expected probabilities yourself, you'll find that that's exactly what you would get. It is important to note that some traits that are not inherited in equal proportions in males and females, they can be sex-linked, but they can also, there may be other things going on, which we'll talk about, I think, next time. So just as a practice question, try to do this problem. Red-green color blindness is a recessive X-linked trait. What phenotypic proportions would you expect for sons and daughters from a cross between a heterozygous mother and a colorblind father? So take a minute to figure that out. Okay, so the mother's heterozygous, the father's colorblind, and what you would end up is with the mother donates 1X to daughters. Half of her daughters are going to be colorblind, half of her daughters are going to be not colorblind, and half of her sons are going to be colorblind, and half of her sons will be normal. So it's important to note that in the small homologous regions of the X and Y chromosome, the pseudo-autosomal regions, anything that's within those regions, those are inherited just like autosomal loci, just like loci that are on the rest of the chromosomes that are not on the sex chromosomes. All right, so the current thinking is that the X and Y chromosomes, the sex chromosomes, evolved from a pair of homologous autosomes, and then at some point some male determining gene, such as in the mammal SRY, appeared on one member of the pair, one of them, and due to some mutation in an existing gene. And crossing over in that region was selected against because that trait is really only beneficial in males. It serves no real purpose in females. And the presence of this proto-Y chromosome would be selected against in females and selected for in males. And so over time, genes that are for male-specific traits start to accumulate on the Y due to natural selection, and they move off of the X again due to natural selection. And the Y chromosome had an inversion event, and this either created or resulted from this palindromic region that currently allows the Y to continue to recombine with itself, actually. And just to kind of let you know, a palindrome is a DNA sequence that is read the same going in the 5' to 3' direction on both strands. So here's the DNA is asymmetric. It has a certain polarity or order to it. Not both ends are the same. We'll talk about the chemistry of why that is later. But so, for example, this DNA sequence reads TGGGAG in the 5' to 3' direction, and it also reads TGGGAG in the 5' to 3' direction on the opposite strand. Okay, so that's what a palindrome is. And this palindromic pattern allows the Y to actually recombine with itself. However, the Y is largely, with the exception of these small pseudo-autosomal regions, it's largely unable to recombine with the X chromosome. So X's can recombine in females, but the Y is just sort of left on its own. It's never going to be with another Y in an individual in a way that will allow it to recombine in any sort of regular way. And so it becomes essentially clonal. It becomes sad and lonely. And over time, it starts to lose regions and shrinks in size. And so the shrinking of the Y chromosome has become stable. It's not continuing to shrink, maybe because it keeps recombining it with itself, and so it's maintaining genetic variation that way, or it just can't afford to lose any more of itself, and any further loss is selected against. So because of this property of the Y chromosome that it is essentially inherited unchanged from father to son, it can be used as a genetic marker to trace patterns of ancestry in humans. And so one example of the use of the application of the Y chromosome as a genetic marker is that it was used to identify descendants of Thomas Jefferson through the lineage of his slave Sally Heming. And so descendants of Sally Heming were claiming Thomas Jefferson as their ancestor, and this was a huge debate between descendants of Sally Heming and descendants of Thomas Jefferson, and eventually they were all genotyped at the Y chromosome, the men were, and were found to be a match for descendants of Thomas Jefferson at their Y chromosome. And so they were able to demonstrate that Thomas Jefferson was, in fact, the descendant of Sally Heming, Thomas Jefferson was, in fact, the ancestor of Sally Heming's descendants. So just a quick note, genetic markers are just variations in DNA sequence. They don't necessarily need to be functional in any way. It's just different patterns of DNA that can be used as a tool to answer certain research questions. And genetic markers can be found within a gene or outside of a gene, or they can be anywhere in the genome. Okay, so the fact that there's one X chromosome in males but two X chromosomes in females presents a gene dosage problem. And what this means is that typically the amount of gene expression is proportional to the number of copies of a gene present, with homozygotes producing more protein than heterozygotes, more of a specific gene protein that's associated with that particular allele than heterozygotes. So essentially, for genes on the X chromosome, females are going to be producing twice as much protein as males are, because females have twice as many X chromosomes. So there can be developmental consequences from having too much gene product or too little gene product. And dosage compensation is a mechanism by which cells equalize the amount of protein that's produced between males and females. And the mechanism of dosage compensation can vary between different species. This is an very active area of research of people who are interested in how cells achieve dosage compensation for the sex chromosomes. So in Drosophila fruit flies, males produce twice as much protein product from their X chromosomes as females do. So the males just ramp up protein production from their X chromosome, whereas in mammals, mammals decrease the amount of X chromosome gene expression by inactivating one of their X chromosomes. So one of our X's in females gets shut down in all of our cells. And then there are some animals that don't have any obvious form of dosage compensation. So either it's not developmentally a problem for them, or they have some way of getting around it that we don't yet understand. So in 1949, Murray Barr observed a dark region in the nuclei of certain female cats. So this is the nucleus of a cell of a female cat, and there's this dark spot that he observed, and these became known as Barr bodies after Murray Barr. And then in 1961, Mary Lyon proposed that these Barr bodies were actually inactivated X chromosomes, and this became known as the Lyon hypothesis. And part of this hypothesis also stated that which X is inactivated is completely random, such that when you have more than two X's present, you get more than one Barr body. And so this random X inactivation takes place fairly early in development, such that women are a mosaic of gene expression on the X chromosome. So this mosaicism is seen in calico cats and tortoiseshell cats, which are usually female. And this patchiness means that this cat is heterozygous for at a gene on the X chromosome that encodes orange or black coloration, and in patches of her skin that produce orange fur, that is indicating that the X chromosome with the black allele was shut down, and vice versa for the patches of black fur. Okay, so individuals with Klinefelter's disease that are XXY, they still have some developmental issues. And so when you have multiple X's, you get as many X's as you have minus one. That's the number of Barr bodies that you end up with. And so it's not known how a cell counts its X chromosomes to know how many Barr bodies to make. It's really, really interesting. But it is important to realize that, with the increasing number of X chromosomes that you have, you get an increasing severity of symptoms, of characteristics. And so X inactivation, even though they're inactivating all of their extra X's, they're still getting an increased effect. And that's because not all of the X is inactivated. X inactivation affects only about 85% of genes on the X, and the remaining 15% remain active. And I just want to say that the gene involved in X inactivation, we're going to talk about that mechanism in a little bit because it's pretty cool in a future lecture, but it's called XIST, for X activation specific transcript, and it produces not a protein but a really, really long RNA molecule. And this RNA molecule coats the X chromosome and recruits other chromatin remodeling proteins to condense the X chromosome and hyperpack it, and this shuts down gene expression along most of it.